In this video, we will be focusing on the pathways for T helper cells, specifically TH1 and TH2. But before we get into all of that, let's go over a bit of background of the immune system and T cells. Have you ever played the game called Among Us? It's quite a popular game that requires strategy and teamwork. The overall objective is for a couple of the crewmates to finish all tasks listed on their screen before an imposter reaches to them and kills them. So let's just say that you are one of these crewmates. You go to do a task in a room called Electrical and see Red in front of you. Just as you are about to turn the corner, Red quickly uses his imposter abilities to kill you off. Now, you are just a body with a bone sticking out of it, as the game would show. Even though the anatomy of these characters in this game is completely different from a normal human, there is one thing that we have in common, and that would be the bone. Though it may look like a normal solidified calcium rock, it actually has a very important function in our body. Hidden underneath all this collagen and calcium phosphate lies the bone marrow, which houses something we call hematopoietic stem cells, or HSC for short. These are the precursors to the T and B cells, which we will be discussing in this chapter. So how do we make these B and T cells? Starting with the hematopoietic stem cells in the bone marrow, these will either mature in the bone marrow to become a B cell, or it will head over to the thymus to mature and become a T cell. Think of it as children growing up. Once they have matured, these cells will head over to the lymph nodes. Let's say now that somewhere in your body there is an infection. That infected area will most likely have a viral or bacterial antigen that cells, which are known as antigen-presenting cells, will pick up and take over to the lymph nodes, where your mature B and T cells are sitting in. I like to think of the lymph node as a university for these mature B and T cells, where they can differentiate or graduate. These mature B and T cells can only differentiate or graduate when they can recognize a specific antigen that they were born to recognize. Once they do recognize their specific antigen, they can receive their antigen diplomas and graduate, aka differentiate, in the scientific term. These differentiated B and T cells are now capable of starting their new jobs, which is to help or head to the site of infection with their specific antigen degrees. And that right there is the basics of how the immune system works. But let's focus on a specific cell in this vast system. When T cells undergo differentiation, they can differentiate into a few different types of T cells. Here we have a cytotoxic T cell, which is also known as CD8+. These cells focus on killing off cells that are considered foreign in the body. They are like ninjas, showing no mercy to these foreign invaders. In order for them to activate, though, they may require the help of helper T cells, which is also known as CD4+. This is the second type of differentiation that T cells can undergo at the lymph nodes, and their role is to help other cells out in showing what antigens are foreign or needs to be attacked, hence the name helper T cells. There is finally a last type of cell that T cells can differentiate to, which are called Tregs, but we won't be covering these cells until future chapters. The main focus for this video is to talk about these helper T cells right here. This T cell will not only become a helper T cell, but can differentiate to a specific type of helper T cell. There are many types that helper T cells can differentiate into, but there are only 3 main types that this chapter in the book discusses. The first would be TH1, which aids macrophages in digesting certain pathogens by sending interferon gamma cytokines to the macrophage and connecting its CD40 ligand to the CD40 receptor on the macrophage. The second type would be TH2, which sends out different cytokines like IL-4, IL-5, and IL-13 to different cells that are capable of fighting helminths or worms. The final type would be TH17, which sends out IL-17 to the neutrophils in order to get them activated to start inflammation in the body. And it can also send IL-22, which isn't depicted here, but it helps to begin production of defensins, which are antimicrobial peptides that help lyse extracellular bacteria. For this video, though, we will only look at the pathways for TH1 and TH2, how these cells are made, and what they do. For the TH1 pathway, the target is fighting off intracellular pathogens, as shown here. These intracellular microbes are detected by two types of cells: natural killer cells and dendritic cells. When these cells detect these intracellular pathogens, the natural killer cells will send out cytokines, known as interferon gamma, and the dendritic cells will send out cytokines, known as IL-12. These cytokines will then reach the antigen-activated helper T cell, which is also considered to be an effector cell because it was differentiated in the lymph node. These cytokines will then induce a signaling cascade that includes the transcription factors TBET, STAT4, and STAT1 in order to get this effector cell to differentiate and drive it into a TH1 cell. Once it becomes a TH1 cell, it will contain the CD40 ligand and it will be capable of creating and sending out interferon gamma cytokines. This is especially important when it comes to classical macrophage activation. When there is a macrophage detected that has ingested these intracellular pathogens, the macrophage may need a bit of help digesting those pathogens fully. When the TH1 cell sees the macrophage holding an intracellular pathogen, they will send their interferon gamma cytokines to the macrophage's receptor and will let their CD40 ligand bind to the CD40 on the macrophage. This allows for the macrophage to be capable of digesting those pesky intracellular microbes, ending this TH1 pathway. This is generally the idea on how these TH1 cells are made and how they help with fighting off microbes in the immune system. For the TH2 pathway, the target is fighting off helminths, aka worms, found in the body. These worms can be detected in the body by mast cells. Just as a side note, they are not limited to these mast cells detecting them, as the eosinophils and dendritic cells can recognize worms too. But for the purposes of simplifying, we will use the mast cells in the diagram. Once these mast cells recognize the helminths, they will send out their IL-4 cytokines to an antigen-activated T cell. These effector cells will take up these IL-4 cytokines in the receptors and undergo signaling cascade. The transcription factors STAT6 and GATA3 will be needed for this effector cell to differentiate and drive itself to becoming a TH2 cell. Once these TH2 cells are made, they will notice the worms in the body and will initiate 3 paths. Just to keep in mind also that there are more paths that the TH2 can take and are not limited to just these 3. Starting with the first sub-pathway, these TH1s will create IL-4 cytokines and send them back to the antigen-activated T cells to produce more TH2 cells. This sub-pathway doesn't really directly affect the worm, but it will help indirectly if more TH2 cells were created. The second sub-pathway that will occur when TH2 cells are created will create IL-5 cytokines. These cytokines will be given to eosinophils, which will take them in the receptors and become activated. Once they are activated, they will create these specific antigens that will latch onto the worm and break it down. Another way of getting rid of these worms would be the final sub-pathway, which is also known as the alternative macrophage activation. This activation requires the help of IL-4 and IL-13 cytokines, which come from the TH2 cell, and bind to the receptors of the macrophage that have already ingested the worm. The macrophage needs more of a boost in this digestion process, which is why these cytokines are needed to be sent by those TH2 cells. Once these macrophages are capable of breaking down the worms with the help of IL-4 and IL-13, this can lead to an anti-inflammatory response and tissue repair, as the macrophage has the worm in its grasp, ending the TH2 pathway. This is generally the idea on how these TH2 cells are made and how they help with fighting off helminths in the immune system.